जैसे कि हमने बात की कि siRNA can be synthesized in the lab and then it can be injected into the cells to perform the gene silencing process. For this purpose, we have to do some homework before we can proceed for the performance of this experiment or this method to design the siRNA molecules. जैसा कि PCR में देखा जाए, प्राइमर किसी भी जीन के लिए स्पेसिफिक सीक्वेंस होता है और उसको हमें डिज़ाइन करना पड़ता है टू गेट द रिजल्ट्स ऑफ एम्प्लीफिकेशन यूजिंग PCR. उसी तरह से कीपिंग इन व्यू द सीक्वेंस ऑफ मैसेंजर RNA which we want to degrade, or keeping in view the sequence of the gene which we want to silent, we need to design the siRNA molecules. The composition or different parameters have to be observed before we can proceed further to use that siRNA to get the required results. For that purpose, we have to look at the number of nucleotides in the siRNA molecule, starting nucleotides, what should be the starting nucleotide of siRNA, then percentage composition of GC content, G और C nucleotide कितने % होने चाहिए, and repeated sequences होने चाहिए या इनको अवॉइड करना चाहिए, and the final confirmation comes from blast search. This is more or less similar to the design of a primer, but here we have to look at some different parameters. 19-mer means 19 nucleotides in a row. जैसे कि dimer means 2 nucleotide, trimer, tetramer, and so on. Similarly, when we talk about siRNA, its major principle is AA 19-mer. That means the first 2 nucleotides should be adenine, followed by the specific 19 nucleotide sequence which is for a particular gene. Then we should keep in mind that messenger RNA, when it is transcribed, it is processed, axonal parts are retained, intronal parts are removed, some modifications comes at 5' end, 3' end, and translation always starts at first AUG sequence, genetic code. And then we have a termination code. Ultimately, we have a 5' UTR, 3' UTR. So, while designing siRNA, we should focus that where the starting point of this siRNA attachment should be and where its ending point should be. As a general principle, the binding site should be between 50 to 100 nucleotides of the AUG start codon. That means we have AUG. From that particular point, 50 to 100 nucleotides, you can have the binding site. And same applies for the translation termination codon. If you have UAG sequence here, that is a termination codon, your siRNA binding site should be 50 to 100 nucleotide before that one. When we talk about double-stranded DNA, one strand is known as a sense strand and the second one is known as an antisense strand. Antisense strand is the one which is used for transcription of mRNA, whereas the other strand is known as the sense strand. Antisense strand has complementary sequence to the messenger RNA and sense strand has the similar sequence to the messenger RNA. इसी principle को अगर हम आगे लेके चलें, siRNA will have a sense strand and will have an antisense strand. So this will be the antisense strand that will bind to the messenger RNA to perform the degradation function. So presence of 2 A's at the start of this siRNA will play crucial role in binding with the messenger RNA and for subsequent function performance. Ideally, the GC content of any double-stranded nucleic acid molecule should be less than 50%. However, for this siRNA experiments, it has been observed that the GC content, if this is between 50 to 60%, successful results have been obtained. But again, different researchers use different percentages of GC content to perform this particular experimental method. As a general principle, 40 to 50% of the GC content, if present in the double-stranded RNA molecule, can be good enough to have no interference or to have no problems while performing that particular method. Then we should avoid the stretches of nucleotides. That means repeated sequences like G, C, G, G, C, C, like this, for the reason that if we have these kind of repeated sequence, whether this is G or C or this is A or T, it can lead towards secondary structure formation when the two RNA strands they get separate. The single-stranded RNA molecule can develop this secondary structure that can be problematic or that can interfere with the binding of the siRNA molecule with the messenger RNA molecule. And the same thing applies to the A and T. So while designing siRNA molecule, we have to pay close attention towards the arrangement of these nucleotide sequences as well and choose the best possible variant. Once the design is done, the arrangement of nucleotide has been observed, then one should blast this sequence of nucleotides using different databases, like for example NCBI database for blast search, exactly similar to that of primer blast, because these sequence blast will tell us that this particular designed nucleotide sequence is a gene specific sequence or it can bind to some number of genes, which is going to cause irrelevant or non-specific degradation of mRNA that will create problem in compiling the results and in claiming that our siRNA experiment has been successful. By default, the NCBI database is going to eliminate any sequence in which more than 15 base pairs have homology with different genes of that particular organism. So blast is the last step of designing, but this is the most crucial step that will determine that how specific is your siRNA to any required sequence of the gene.